We'll get started with this. On the first slide, the only thing I want to show you here is the email address. This is the best way to contact me. If I'm not able to answer your questions or get to all your questions, I look forward to, through email, getting back to you, and I'd be happy to do that. Today, what I want to discuss are three different areas. I'm going to briefly touch only on the first area, and that's the patient-derived xenograft models that are established by repeated subpassaging. And these are used to preclinically evaluate tumor responses to therapies that are targeting the tumor and not the tumor microenvironment. My interest is focused largely on the area of PDX models that are optimized for establishing microenvironments. So in the second area, we're going to talk about the logistics and plannings of PDX models that are involved in microenvironments and how we use them. These have all been published, and I'm going to talk about them just to show you examples of some of these are still being used, but I'm also going to tell you some of the setbacks and the deficiencies of these models. The other thing I want to do, and I'm going to spend much more time on this, is to introduce a relatively new model called the OTX model, or the mental tumor xenograft model, and I'm very excited about this, and I want to spend a lot of time on this because for some of you in the audience, I'm hoping will be able to pick up and use this model, and you should be able to do that after you listen to my last part of this session, because it's a very simple thing, but a very powerful way to look at tumor microenvironments and to investigate their immune suppression and ways to overcome this suppression. So to begin with then, I'm going to just walk you through some of the PDX models that I talked about. They're derived from these patient xenografts that are just involving subpassaging of the tumor. And here we start with the patient's tumor. The tumor is in red and the microenvironment's in blue, just for the cartoon. You start by engrafting these mice in the primary recipients, and this is usually just by putting pieces of tumor underneath the skin and then subpassaging them after they've established and they're growing logarithmically in the mice, and they go on to the secondary recipients. And you can see here that the tumor microenvironment is starting to be replaced by the mouse microenvironment. These are mouse stroma, but the tumor itself, hopefully in red, is maintained in a stable fashion, and that can be tested here by looking at gene expression patterns in the mouse and also comparing them to what originally were seen in the patient. And then we go on to tertiary recipients, and the whole reason for doing all this is to establish a large number of mice so you can study multiple cohorts and look to see what effect the therapy has on them. A good example, I'm just taking a couple of these examples so you can take them from the JAC's website, but this is ovarian tumor PDX responses to a standard therapy, first-line therapy of patients with carboplatin and paclitaxel. And as you can see here, this treatment was able to suppress the progression of the tumor. But the real value of these is hope that these PDX responses will mimic that which will be seen in the patient, so that you can either have a concordance of these with knowing what's going on in the patient and what's going on in the PDX model. The hope is to be able to predict the response. I'm going to show you just one example here. Starting here, we have a person that failed the first-line chemotherapy. They were then treated with erlotinib, which is a kinase inhibitor at axon epidermal growth factor receptor. And you can see that when they were treated with that, the tumor burden went down, but then it came back up again. And at this point, there was a thoracotomy done, and the thoracotomy released tissue that could be used to establish the PDX model. And so a number of mice were implanted and subpassaged, and these mice were then used to monitor the treatment of a variety of different approaches. But the one that worked in the patient, which was serteximab and afatinib, this caused a decrease in the tumor progression. As you can see, it also, there was a concordance in the mouse PDXs that there was also this suppression with this particular combination. And this is ideal. It's not always happening, and it's not. There are also some problems with this, which we're going to discuss in a little bit. But I wanted to end with this because there's a lot of companies in the United States, as well as in Canada and Europe and Australia, I imagine, where they have the PDX programs. And you're welcome to go Google these companies and talk to them about their models, because they have models that you can take off the shelf and deliver to you as mice or as frozen samples, and these are very useful. This is not something that I'm working with, and I want to be able to talk about something else here that I am interested in. But first, before we go on to that, I want to say that there are certainly advantages to the passaging of tumors to look at this. We've just discussed a couple of them. You're able to generate over 1,000 mice bearing the tumor xenografts and more. It's unlimited. We have here, we can compare the gene expression patterns that are included, copy number and variations between the original tumor and the xenograft tissues. And there's a whole host of other examples we can give, but those are advantages. But there clearly are disadvantages. This requires several months, or at least a month, probably more, to establish. So if you're looking at a patient, you want to start trying to predict what's going to happen to that patient. This is going to take a while, especially in these resistant tumors, and these patients don't usually only have a lot of time left. So this is a problem. There's also a loss of most or all of the stromal cells after, I found, even after the second passage, but certainly into the third, fourth, and fifth passage, you're starting to lose stromal cells. And if you're interested in knowing what's going on in the tumor microenvironment, you're not going to be able to do it with these models. So let me begin then by introducing what I think is a way to look at the entire tumor. And this is an alternative. It's not necessarily better, but it's different. And it depends on what your question is. And I'll get to that in a minute. But these are designed to optimally establish and study the human tumor microenvironments. And with it, this will, as we know, and as you all know, I'm sure that the tumor here in red is more than just tumor. It's surrounded by a whole host of other cells, these we call the stroma. And this stroma includes the fibroblasts. It includes the T and B lymphocytes, the CD4 and CD8 lymphocytes, the whole host of lymphocytes, the plasma cells, and macrophages and monocytes, and also blood vessels. So clearly there's a lot going on here, and these cells here are interacting with the tumor, and the tumor interacting with these cells. And the next slide just shows us the actual setting. And here we have a tumor. This is a non-small cell lung tumor, but all these tumors, the solid tumors, are virtually similar to this. You see very healthy tumor, and you see collections of inflammatory leukocytes. If you go over here to the higher power, you can see these lymphocytes are present. They're actually plasma cells here, and we've actually looked at these cells both by flow and show that the majority of the T cells, and I'm sort of focused on T cells, but the majority of T cells have an effector memory phenotype, and these cells are hyperresponsive. We and others have shown this for a while, that the T cells that are present in the tumor microenvironment are hyperresponsive to activation via the T cell receptor. And for that reason, they don't seem, while they're in juxtaposition to the tumor, they're very close to the tumor, they don't seem to be controlling the tumor. And we're interested in knowing why. We'd like to know why these cells are hyperresponsive. I call it hyperresponsiveness. Other people have terms like tolerance, and we won't get into that. But these are just hyperresponsive to activation. And this is reversible. If you take these cells out of the tumor microenvironment, they become functionally responsive again. So that's our question, and we'd like to know why that is, and also are there ways in which we can reverse the function of these T cells. And to do that, we've turned to our models, the PDX microenvironment models. To begin with, and this is just historical, this slide I've thrown in because it goes back a long way. This is some 28 years ago, was the first successful engraftment of a tumor into a SCID mouse. There's a lot that's happened since then over the last 30 years or more. And some of it's been good. I mean, we've really improved on our models, but along the way, all of us, including me, have been so excited about that. Sometimes we've oversold the model and it's gotten a bad name. But we are now hopefully putting aside the hyperbole and maybe focusing on actual results. So along the way, I'm going to tell you what we've done, but I'm also going to tell you about some of the deficiencies of the model and how we might overcome them. So to begin with, I think it's important, I have a mixed audience here, and so any of you that are well involved in this, just bear with me, I'll get on to other things. But I want to start with a little conversation about the logistics that are involved in generating successful PDXs for the study of microenvironments. So I want to start with the logistics, and we'll start with daunting of all the logistics, and that's to get started. You need to get a tissue procurement set up. And this involves not just going to a surgeon and asking him to pass you some tissues. You really want to get the surgeon and the pathologist on board with you. You want to make them part of your team. You want them to have a vested interest in what you're doing, because you're going to depend upon them, not just for giving you the tissues, but the surgeon is going to tell you how his patient is doing, how he's, even though this, you're not going to know the name of the patient, this is going to be anonymized because of IRB restrictions, but the surgeons will be able to tell you that what's going on, what he's treated the patient with, and he's going to want to know what you're finding out in your study. The pathologist is absolutely essential, because that pathologist is going to give you quality control of your tissue that you're getting, and he's going to tell you the histopathological diagnosis. And the other thing, the sine qua non of all tissue, it's got to be sterile. You don't want to insert or implant non-sterile tissues into these immunodeficient mice. I think you can well figure out what might happen there. With regard to tissue preparation, there's a variety of things that we do. We start off, and what I'm going to talk about are actually pieces of tumors that are not disrupted, so we can actually implant a tumor microenvironment and expect that to sustain itself. We also are injecting, and I'll talk about this at the end in the mental tumor xenograft model, that we're injecting cell aggregates, which are not actually pieces, but they're actually aggregates of cells that will generate the tumor microenvironment. Other people are injecting single cell suspensions, and these all can be either fresh or frozen. Now, the other thing to be considered, of course, is the implantation. This can be done by either subcutaneously, and we don't use trocars. We actually open a piece of skin and implant the pieces that we want to implant, and we close it with the surgical adhesive. You can also inject IP, as I mentioned, and IV with single cell suspensions. And of course, there are orthotopic implants. People are injecting tumors, lung tumors in the lung, and bladder tumors in the bladder, etc. The next couple slides I'm going to show you, I'm not going to go through. I just want to let you know that this is what we do, and you might take a look at these later on to see the logistics of what we've set up in terms of both the tissue procurement, which has a lot on this slide. I'm not going to go through it. I'll come back to it in a minute. But the other thing is we have a flow chart for the processing of the tumor, and we're getting ovarian tumors that I'll be talking about a lot. We're getting ovarian tumor, solid tumor tissue, as well as the ascites, and we go and have a definite plan for doing this. And this is something you want to set up well in advance. And I want to go back just for a minute to go over something that happened, that is on this slide, that happened to me yesterday. I got a call at 3:30 in the afternoon from pathology. The pathologist had gotten the tumor. The tumor had been received by pathology. The pathologist was looking at it, had made his diagnosis, and he's ready to give us tissue. That tissue was delivered to them, and I got a call saying, I've got a 7-gram tumor. This I was delighted about. I said, this is wonderful. We can now inject up to 70 mice with this, and we'll, with our new technique, we only need to inject 100 milligrams of the tumor tissue, the wet tumor tissue. And then, so I was ready to dispatch somebody to go down and pick it up, but all of a sudden the pathologist called me back and says, well, Dick, it's not going to be something you want, because we found out it's about 60% necrotic. And he was right, and I turned it down. But this is why it's important to have yourself in place, a team of people who are going to be able to oversee the tissue and tell you what to expect. It's garbage in and garbage out. You don't want to put in necrotic tissue, obviously. So I'm going to begin just with one of our examples of the tumor xenograft. And this is an engraftment of non-disrupted tumor biopsy tissues that we get. It's fresh tissue. This is mostly done fresh. We have done it frozen, but what we see here is we take a tumor. This is a non-small cell lung tumor. And what we do is we're taking this out and implanting it subcutaneously. And our question was, can we reactivate those T cells that I was talking about? And the way we've done that is we've made biodegradable microspheres that have, we've included IL-12. The control microspheres that we're injecting were with BSA. And this actually was implanted in the ventral midline in around the mammary fat pad. But basically, what is found then, we go and want to evaluate the effects of IL-12 by monitoring the activation here and the serum levels of interferon gamma, because if there's T cells there and they are responding to IL-12, the T cells are going to start producing, as well as NK cells will start producing interferon. Interferon has an effect on hundreds of different genes, and we're going to monitor some of those changes as we go forward. So just to begin with, the first thing we're looking at, the easiest thing to look at here, is the fold entry, or this is the the increases in interferon gamma that are measured here as average levels of increases. Each one of these is a different mouse. All but 2 of these mice did respond by a significant increase in the interferon levels, and this was at least indicating to us that there was a reaction going on. The next thing we look at is histology, and I want to point out a couple things here with this. These are the controls, and as you can see, just 5 days after the control, the tumor is growing very well. There's actually evidence of mitosis here, that these are healthy-looking cells, opposed to the control where the mice were treated with IL-12, and these IL-12 microspheres, I should say, were injected directly into the tumor microenvironment to get a local and sustained release of IL-12. And we're already seeing an increase in the proliferation of the lymphocytes, and there are also apoptotic bodies here that are starting to show up. And the other thing I want to note is that if you look just at the bump on the skin, the size of the bump that a lot of people use, there was no difference. There was no statistically significant difference in the size of the bumps, whether it was treated with BSA or treated with IL-12. And that's important, because you're going to be fooled on occasion if you're just using, the only thing you're using is a volume of your tumor, because that volume may indicate tumor is growing. It may also indicate that there's an inflammatory response to the tumor. Now here's a look at what the tumor looks like 14 days after the treatment with IL-12. And as you can see here, in the BSA treated, the tumor's growing. There's a lot of fibrosis here, but there's a healthy tumor. But in the IL-12 microsphere treated group, the tumor has been totally effaced by a proliferation of the lymphocytes, and larger view here. And these lymphocytes, we were interested to see, well, what kind of lymphocytes are they? Are they B cells? Are they T cells? Are they memory cells? And the like. And so to do that, you can just simply look at the immunohistochemistry of these, and they are CD45. This is human CD45 cells that are present there. They're CD5RO, so these cells have already been activated. I'm not going to show you a lot of immunohistochemistry, but just to show you, they're all the T cells that I'm focused on and I'm interested in. But in addition to that, we do see the CD20 B cells and CD138 plasma cells and the CD68. So we do have a real response that can be monitored and charted, and not just looking at the bump, as I said, bump on the skin. So looking at gene arrays, and I'm not going to take you through all the gene arrays, I just want to give you a conclusion that we have. As I told you, that most of these are a consequence of interferon. The interferon will induce a whole host of things that go on, but our conclusion from this was that the IL-12 here, the IL-12 is inducing an upregulation of genes associated with both the innate and the cognitive immune responses, and it induces decreases in genes associated with angiogenesis. This I'm going to summarize for you in just a minute, but I think rather than go on the details of this, it's just what can be done and what is being done. I want to go to a cartoon here and just tell you this is what we started with, the tumor microenvironment. As I told you, we perturb that microenvironment by injecting these biodegradable microspheres with IL-12. They release their IL-12. That IL-12 is going to then has stimulated the proliferation of T cells, and these T cells keep proliferating. They eventually will produce interferon. That interferon will act on macrophages and other cells to upregulate iNOS and produce nitric oxide, which will be toxic to the tumor as well as to other cells, of course. And other things are happening. I'm not going to go through but we're very interested in interferon upregulating IP10 and Nig. These are chemokines that will attract other T cells into the tumor microenvironment, which we're not looking at, but we're looking at these actually holding these cells in the tumor microenvironment. And these chemokines, as well as other chemokines, are involved in anti-angiogenesis, which is in part what's going on here, I think, in terms of the eradication of the tumor. We are shutting down a blood supply. In this case, it's a mouse blood supply, but it is shutting it down, and these tumors are going away. They're being eradicated, and they're being overcome with fibroblast. The question of these fibroblasts in this particular model are mostly, at this point, they're mouse fibroblasts. These cells all around the tumor, the lymphocytes and macrophages, etc., these are human, as I've shown you. There are disadvantages of these early SCID models, and I mention this because these models are still being used, and I'm not trying to tread on anybody or overly criticize, but just to keep it together here, the host versus graft response, These skid mice and also non-skid mice, they still have an innate immune response. And this ultimately leads to the rejection of the xenografts, and it sometimes makes interpretations difficult. The tumors also, I found anyway, they don't typically metastasize when they've been injected subcutaneously. Some do, some don't. The xenografts are slow to develop. It's over a month, and I tend to be a short-run hedonist. I like to see things rapidly. The stroma in the xenografts are replaced by mouse cells eventually, and we'll show you how that happens actually within a matter of 6 to 8 weeks. There are other problems that have been reviewed in a paper. If you're interested, you can go and look at that. But there's a lot of disadvantages that we have to deal with, and we're looking to try to overcome those disadvantages, and some of them have been overcome. And most of the advances recently in tumor models, these particular microenvironment tumor models, have come with modifications of the immune deficient mice. This has been undertaken by a lot of people, but Lenny Schultz has a wonderful review in Nature Reviews in Immunology if you're interested. And what he's found, and I want to focus on, is just one. This is the NOD-SCID IL-2 common gamma chain receptor knockout, commonly called the NSG mouse. Many of you are familiar with this, I won't dwell on it, but this mouse is very immunodeficient. It doesn't have an innate response, it doesn't have adaptive response. And we and others have looked at this. Early on, we found that there was a long-term engraftment, and I'm going to show you a couple examples of this and how this worked better than the SCID mouse. But we found that we could maintain the tumor microenvironment for 9, 10, 12 weeks, even longer. And this was really a very exciting time for us and others, and now people are largely moving to this model. And I want to show you just an example. We start again by injecting, this is where we started off in these NSG mice. Again, we're just injecting the small pieces of these tumors into the NSG mouse, and we're monitoring out 12 and actually going on to 20 weeks. And we're looking again at treatment, and we're looking to see whether we can immunostimulate the T cells that are present in there. And I've already shown you that we can do that in the other model. We can do it in this model too. But I wanted to show you something else, and just to compare the results that we see histologically with the tumor of comparing. Now what we're doing is we're going to see here, this is the tumor before it's implanted, and here it is 9 weeks after an NSG mouse has received the tumor. Again, we can stain it with cytokeratin or EpCAM or other tumor markers, and there's tumor here in the original, of course, and at 9 weeks out, there's still tumor. I don't think we've done a lot of staining with this, but I just wanted to show you here. This is Ki67, which will show you that there's these lymphocytes, the leukocytes actually are dividing here, and so are also, there's evidence of division of the cells in the tumor itself, and the same with the engraftment. If you get down to the lower panels here it is again, it's just showing that the tumor is still there in the NSG mouse, but look what's happened to the tumor after 9 weeks in a SCID mouse. The tumor has actually been totally eradicated or effaced by fibroblasts and other cells. And if you look at HLA, staining with HLA, most of the xenograft in the NSG mice are still human cells. There are some mouse cells, because if you look for CD45 over here, there's some CD45 mouse cells present getting into this tumor. But look at the xenograft here, where the xenograft was. It's largely infiltrated with mouse leukocytes. So there it is, and here's something I wanted to point out in this model that I was totally surprised about. But what we found was that the human T cells and plasma cells relocate from the xenograft. We have these are CD3 T cells, but look, they're moving on by 6 weeks into the spleen, and there's just a whole host of them. There's a plethora of these T cells there. They're in the lung, they're in the liver, they're in the kidney, and they're in the intestine. I was intrigued by this because we want to study, we want to do biochemistry on these T cells, and so this provided us with an opportunity. I'm somewhat off target here, but I wanted to show you this, and we can actually take these cells and we can transplant them into another NSG mouse, and these cells will take off and survive, and that's shown in the next slide, I believe. Yes, these are tumor-associated T cells from the spleen that were injected into a non-tumor-bearing NSG mouse, and you can see here we have these cells. We can then do flow analysis on them, and we can actually phenotype them and show that these cells are expressing markers that are consistent with an effector memory T cell. And interestingly, these cells are no longer immunosuppressed. They've left the tumor microenvironment, and that immunosuppression is reversible, and that information was useful to us. Some of the disadvantages, though, of these NSG PDX models where it still is taking us a month or more to establish here, and it doesn't mimic the exact progression, or it doesn't mimic the patient's progression, like with metastasis and ascites formation that we see in the ovarian tumors. And there is an eventual replacement here. It starts to lose the human stroma, and you are getting some mouse stroma. So I'm going to slow down now because I'm getting to something that I think is new. Some of this has been published, some of it's unpublished. Most of the utilization this has been unpublished. But yeah, I want to just give you an example of what we're looking for in an ideal model. We don't have an ideal model necessarily, but we're approaching that, I think, with this new model that I'm going to introduce to you. And in this model, we have the first thing I would like to see it develop rapidly. I would like to do it in hours, but if we can do it in days and weeks, and that's the ideal. The other thing is we want long-term survival of the tumor and the stroma so we can evaluate it going forward. We want the ability to be able to recover and quantify and phenotype and assess the function of tumor and tumor stroma. And that's going to be done in this next model. We want to demonstrate the immune suppressive nature of the tumor microenvironment. I'm going to show you how that's being done in this new model. We want to be able to evaluate the ability of immunotherapeutic strategies to overcome immune suppression, and I will show you how that's being done. And we want at best to mimic at least the patient's tumor progression, and hopefully eventually to show that the responses mimic the patient's responses to therapy. With that in mind, I'm going to introduce to you the mental tumor xenograft model. I think Lenny named this. I got to talking to him about it. He says, oh, call it the OTX model. Okay, it's the OTX model. I don't want to take full credit for this. I don't want to take any credit for it. I just want to open this up to you, and hopefully that you may agree with me that this is something you might want to try. And that's why I'm going to walk through this slowly and somewhat methodically so you can pick up and do this. And I apologize. I'm going to probably be redundant, but my attempt is to entice you into trying this. And it's a very simple procedure, and it's a very powerful one, I believe. But as I said, this was not my idea initially. This came, to me anyway, from Scott Gerber and his colleagues John Freilinger and Edith Lord and others in Rochester. They published a couple papers. But what they found was that they took these murine tumors and injected them into intact mice. These tumors, single cells, would go preferentially and attach into what is called the greater omentum. And the sites of attachment exhibit a unique and vascular microenvironment, which they showed, which appeared to be supporting the growth and survival of the tumors. What I'm going to show you next is just a picture of this, and we'll show more of this later in a video that we'll be showing you later, but this is the omentum. It's the greater omentum, and it sits on the greater curvature of the stomach, and it's a very small, it looks large here, but it's blown up. This is only about 1.5 centimeters long. It's very thin, and it is in parts translucent. And it has, in intact mice, it has, if you can see these, I can see them, I expect you can, these white spots. They're called milky spots. They're just loaded with lymphocytes. And the thing is with this tissue is loaded with blood supply too, as you'll see, and it's also loaded with adipocytes, which are just fat cells. So with that as a backdrop, I want to go on to some other things that led us to establish the xenograft model. And this was the observation of surgeons make, and they have shown that the peritoneal tumor metastases often adhere to the omentum, and this is in epithelial ovarian cancers, which we're now going to focus on, and we are focusing on. It occurs with pancreatic cancers, and it will occur with after tumor resections with gastric cancers and colorectal cancers. So here's the omentum, the greater omentum in a human. You can see here is an example of a tumor that's metastasized there. So these tumors, when they break free, they really reach out and appear to selectively home to and set up housekeeping in the omentum. The other thing I mentioned, there was a paper that came out about this time in Nature Medicine, and it was a wonderful paper which showed that the adipocytes actually promote ovarian cancer metastasis, and these adipocytes are fed upon by the tumor cell. They actually have receptors for fatty acids, and this is a source of energy for the tumor. So with all this in mind, we set out to see what would happen if we injected tumors. And there may be people who have done this already, and I maybe find that out from the audience. but I'm not aware of it. But what we did was we simply took and injected intraperitoneally injection of fresh or frozen tumor cell aggregates. These were derived primarily from human ovarian tumors, solid tumors, but we also have done lung and B-cell lymphomas, but I'm not going to talk about that because we haven't done enough of that to really be consistent and tell you what I know. What I know is going to be with the models for ovarian cancer. You inject them into the NSG mice, that results in a rapid, I will tell you rapid, and we've actually targeted this, it's not just days, it's within hours. They preferentially home to and engraft within the xenografts, the greater omentum, and also in the lesser omentum, which is right around the corner. And this reflects, these xenografts actually reflect the histological characteristics of the tumor microenvironment. I'm going to show you that. The omental xenografts can be removed, enzymatically digested, and then the tumor cells and tumor-associated lymphocytes can be recovered, quantified at the single cell level, phenotyped, and assayed for function. And we're going to go on from there. Well, this model was been published in Cancer Immunity and PLOS ONE. A lot of what I'm going to tell you right now in the utilizations of this have not yet been published, but I want to show you to entice you anyway. So here we're looking at just the aggregates, the tumor aggregates. They're stained with H&E, stained with CD45, showing that there are leukocytes there, stained with CD3. There are T cells present there. We stain with trichrome, and there's evidence of fibroblasts there, and there's evidence of the tumor staining with cytokeratin. So the way we generate these aggregates, we just put them through a stainless steel wire mesh. It has about 0.3 millimeter, a little bit over, maybe about 0.4 millimeter holes through the mesh, and what comes out are these aggregates. And this is purposely so that we can maintain an intact microenvironment. And it's these aggregates that are moving to, preferentially to the omentum. I have now something that Tony Saka in the lab has put together. It's a little video, not a video, it's a little cartoon, what's going on. And I'd show you this. What we do is we load a syringe, and actually I want to tell you, and Tony has told me, you want to make sure that in the syringe these aggregates remain in suspension, so you keep going back and you're only going to use maybe 2 or 3 times into 2 or 3 mice and then go back and get some more. But this is then injected intraperitoneally simply into the mouse, and these cells will then, as I said, they will home to, and they will then go into on the greater curvature of the stomach. That's the stomach, and on its greater curvature by day 1 and 7, the tumor cells localize preferentially here into the omentum. I say that this is where we were looking at, but when we started looking within hours, we're seeing this happening. So it's happening very rapidly. And we'll see within a week to 2 weeks, we're actually seeing the tumor and the tumor microenvironment. And here you just see an H&E of this tumor microenvironment. And I wanted to mention here, since this is a very thin organ, we can actually take it out in its entirety. We can stick it in a tube, and we can stain it. Here we're staining with Fitzy EpCAM, which is for the marker, for a green marker. Here we have for the tumor. We're staining for mouse vessels. The human vessels start up, and it's really taken over by mouse vessels, and these are mouse vessels here, and they're very tortuous. And we also can stain with human CD45 to show that there's human leukocytes present in there. And as we progress, after 14 days, and this sometimes is variable, it's maybe 2 to 3 weeks later or further on, we'll start to see a dissemination of the xenograft from the omentum into organ sites in the peritoneal cavity. So it'll move from the omentum here, it goes into the ovary, and we look at the pancreas, the uterus, the spleen, and the liver. But it's at this point, it's still primarily located within the peritoneal cavity. And at any rate, this is where we are with this. And if we let this go, what happens is we're going to be getting the metastasis eventually out into the diaphragm. As you can see here, in the diaphragm, there's tumor, and in the lung, there's tumor. And if we wait, and this happens very long, and it happens very often in the patient as well, we get an ascites fluid, which is CA-125 positive. So this really mimics the pattern of what we see in a patient. We see local growth of the tumor, it spreads throughout the peritoneal cavity, it'll break through, go into the lung, and we'll end up with an ascites fluid. So I'm going to be moving along. I'm just going to review here just to say the same thing. I showed this slide because I wanted to emphasize that we've looked at the optimal dose of the wet weight of the tumor cell aggregates we're injecting, and that's right around 100 to 150. And with that, we have an ability to inject a large number of mice. We can actually go to 50 milligrams of the tumor tissue, and it just takes longer. If we go, you think a little bit is good, more will be better. Well, that's not the case. If you overwhelm the omentum with this, you end up with necrosis, and it's not a good engraftment. So we have an optimal level of dose. And we just, this will go into the omentum, and then we can go and do our things. And this is just a histology to show you again that these tumors are very viable. They're mitosis here. And notice they're right in juxtaposition to blood vessels, both the tumor and the lymphocytes, and they're very happy and comfortable here. This is just more showing you the histology. These tumors are very well spread throughout the body after they've left the omentum. And in here, we just show that there's, on the diaphragm, we do see a tumor, and we see the lung, in the lung as well. So I want to end this whole thing with this will go through fairly rapidly, but because I haven't got a lot of time left, but I want to let you know that we can quantify and phenotype tumor cells and lymphocytes present in the tumor xenograft. We can demonstrate here the immune suppressive effects of the microenvironment, and we can determine the mechanisms by which T cells and CAR T cells become arrested in this tumor microenvironment. And we can monitor changes in the vasculature, and we can preclinically evaluate chemo and immunotherapies. This is a standard treatment, and I won't go through it, but it's just the way to digest your tumor with enzymes, and we have other ways of getting the cells out, but this is a standard way of recovering single cells from a tumor xenograft. You can see here we have isolated here. We can isolate and phenotype, and by flow cytometry, we can identify the human T cells, the human, in this case, CD45 positive cells, and we can see here that we can isolate and detect the EpCAM positive tumor cells. And this just is showing you here that these are individual tumors from a mouse out of the omentum 6 weeks after they've been injected, and we can quantify and detect the actual numbers, the absolute numbers of these cells in individual mice that we show here. One of the things that this model is unique for, I think, is that when we inject the lymphocytes, IP, they will localize rapidly and preferentially into the tumor xenograft. We've determined that this is very useful because we can now adoptively transfer in T cells that are perfectly functional, and we can actually adopt and adoptively transfer in the CAR T cells as well. And I'm going to show you the consequences of that. So if you adoptively transfer in the T cells, I'll tell you the message here and then show you, but these CAR T cells, they become anergic or hyper-responsive following their entry, shortly after their entry into these tumor microenvironments. Here is just one slide I'm going to show you that shows that the endogenous cells are hyper-responsive. You can see here that there is a downregulation of these compared to a normal activation. We should be up here with normal activation. This is just immediately after the injection and entry into of exogenous cells into the tumor microenvironment. There is a very robust activation seen here, and after 3 days there's still a robust activation, but after 7 days you can see we now have back down on the response, and these are hyper-responsive just like the endogenous tumor-associated cells. This has also been shown, others have shown this, people at University of Pennsylvania, that tumor antigen-specific CAR T cells, their function is reversibly arrested once they enter the tumor microenvironment. This was done independently by them. For those of you that may not have been keeping up with what's going on in the field of cancer therapy, most of you probably have, I'm just going to show a cartoon of the CAR T cell, and this is a CAR T cell that has been put together. We have a single chain fragment variable region of VH and VL of the immunoglobulin that is tethered to a spacer and a transmembrane, and the intracytoplasmic domain of the signaling possibilities or the signaling molecules of the T cell receptor, and then we're off and running. So we can now, by doing this, we can convert patients' T cells into cells that will recognize the tumor, and they do it recognition by how an antibody was in the absence of MHC restriction. So I'm going to go through, we're running out of time, and I want to just show you this, but what the people, Ed Moon and Carl June and Steve Albelda and their colleagues at the hospital at the University of Pennsylvania have shown that these T cells are hyper-responsive to killing their target cells, and if you take them out and incubate them in vitro, this is reversible. So they're suppressed in the tumor microenvironment for killing. They're also suppressed for when they see their tumor antigen, they're suppressed for interferon gamma, but this is reversible. The next slide shows that these same investigators were able to show here that we have the PD-1 blockade, but what I was interested in, diacylglycerol kinase inhibition restores their activation. So we now have a way, here is with the diacylglycerol kinase, you can see that both diacylglycerol kinase inhibitor 1 and 2 will reverse this. I haven't got time to go into the details, but this is a way to reverse this, and it's something that can be a target to be used clinically, which is underway, I'm sure, by them and by us. We're looking at this. I'm going to try to finish up quickly with the OTX model that led us to determine what was responsible for the T cell and CAR T cell arrest, we think, but it's certainly with the T cell arrest in the microenvironment. And what we observed, and I'll just want to tell you this, we found in the tumor ascites and in the solid tumors, we find extracellular vesicles by ultracentrifugation. We can recover these vesicles, and these are 7 to 150 nanometers, and we can actually induce a reversible energy in human T cells with this. I haven't got time to go on with this. This is a manuscript that has been submitted to this. If you're interested in more about this, we can talk about it, but here's an example. Here's just with ascites fluid, the tumor ascites fluid, whether it's from the patient or whether it's from the xenograft, will suppress the activation. And here are the extracellular vesicles derived from that ascites fluid, and which has shown that you can shut this down. And interestingly, we can reverse the arrest with diacylglycerol kinase inhibitors, both here and partially reverse it here. So strategies are now being tested to look at and preclinically evaluate chemotherapy and immunotherapy. This is much a work in progress, but this is an interesting early observation we've had, is that if you add chemotherapy, which is the standard carboplatin and paclitaxel, you see here these mice survive well beyond the controls that are now already all dead. In the same series of dots here are the chemotherapy plus IL-12. And so there's no death, there's no development of ascites syndrome, and we don't see any obvious evidence, grossly or of the tumor. But if we look down here, the mice that were treated with the chemotherapy only, we've identified micromets into the lung and also present on the diaphragm. This is very premature, and we now have a whole large cohort of mice being studied to look to see whether these responses are paralleling the responses we see in mice. This is done blindly, and I'm not free and able to tell you the results of that at this point. So I will end here just with a very quick statement that one thing that we haven't taken advantage of, but we can actually quantify changes in the vasculature. Even though it's mouse vasculature, we can see that after an implantation, very soon after an implantation, we see here's a control up here, vessels, and here are these large plethora of vessels that are very tortuous and can be quantified. And here's with tumor and fibroblast, and I am not going to go into this right now. I just want to end with a summary of the OTX model and its uses. As I said, we take the tumor, we make the single cell suspensions that is implanted in here. We can look, and I've shown you the immunohistochemistry of this is established very quickly, and it's also durable. We can get it 19, 20 weeks out. We can also recover and assess the function of the T cells. We have the whole mount evaluations of what's going on in terms of the tumor, the vessels, and the leukocytes. And we can also enzymatically digest and quantify at the single cell level the tumor as well as the lymphocytes here. I'll end with these conclusions at this point. What we have is, you can read this for yourself, and I won't go over it, but I think I've belabored the point. I hope that you at least some of you will consider using this model, and if you have any questions that can't be answered today, that you'll fax and you'll email me your questions, and I will get back to you and help you out with this. The last slide, I want to acknowledge from my lab 3 people who were very instrumental in the development of this model and its use. And the group is headed by Sandra Yokota. Bob Parsons has been outstanding in his monitoring of the mice and helping me out with this, and Tony Saka, who has not only been instrumental in the development of this mouse model, but he's also helped a lot with putting this whole talk together with the people at JAX, and I want to shout out to them. Megan McCauley and Kathy Snow have been really very helpful, and it's greatly appreciated. People at Roswell Park Cancer Institute, Paul Wallace in the flow facility, Dr. Kunle Odunsi, he's just been an outstanding collaborator. He is the surgeon and physician scientist. We've had help with confocal microscopy here at UB, and my long-standing collaboration with Lenny Schultz. None of this would have been possible without him. I'm running a little bit over time. I'm going to stop here, and we'll get into, I think, the question and answer portion.